Gummy. Today I will be presenting on a paper called Application Potential of Bone Marrow Mesenchymal Stem Cell-Based Tissue Engineering for Spinal Cord Defect Repair in Rat Fetuses with Spina Bifida aperta. Or much simply put, the promise of stem cell-based treatments for spina bifida. So why is this research important? Spina bifida is basically just a neural tube defect in which the neural tube protecting the spinal cord fails to fully develop. So this occurs in the second trimester of pregnancy. Anywhere from around 16 to 26 weeks, this happens in the fetus. So just to give you a better visual of what this neural tube is, this is the neural tube. In early development, that would be the head, and this would be the spine of the fetus. And you can see in this image that it starts to kind of fuse together. But in spina bifida, it doesn't fuse all the way. Now this gap in the neural tube creates a number of complications for the fetus. So in the least severe form of spina bifida, called spina bifida occulta, this kind of break in the spinal cord isn't exactly significant, but it is a loss of protection for the neural integrity of the fetus. In the next severity of the defect, this break is a lot more significant. There's a lot more space in between these two folds, and in that occurrence, there's a buildup of cerebrospinal fluid or brain fluid that puts pressure on the neural tube. Lastly, what my paper is addressing is myelomeningocele spina bifida. Now, this is just when meninges or neural matter is incorporated in the cyst, and that definitely compromises the neural integrity of the fetus. So this is a real-life ultrasound of a fetus in the third trimester. This red arrow is pointing this to the myelomeningocele. You can kind of see the outline of this cyst, and these exposures of light are actually the meninges, or the neural matter. So what are the current treatments for myelomeningocele? There was actually a study performed by the New England Journal of Medicine a few years back that assayed the efficiency of perinatal surgery. So this is just a procedure done either a few days before birth while the baby is still in its pregnant mother, or a few days post-birth. So what they found was that it minimizes fluid entering the brain, which is actually called hydrocephaly. In this case, there would be pressure being put on the baby's brain, which causes a ton of brain defects or a ton of brain complications. And it also gave the infant an opportunity for a normal life, but I'll talk about why that's kind of ambiguous. The major con of this procedure was that it actually induced premature birth in a number of the infants or fetuses that it was performed on. And a key thing in the gestational period or pregnancy is to maximize the time the baby has to incubate inside its mother. So that led to a lot of complications like incontinence, paralysis, and even chronic pain. So yes, these surgeons are giving these babies an opportunity for a normal life. They're expanding its lifespan, but what is that baby looking at? It's looking at a life of chronic pain, of paralysis, of constant dependency on its peers. So before we get into my paper, I mentioned stem cells before. Let me just give you a quick rundown on what they are. Basically, stem cells need to be able to perfectly replicate and perfectly differentiate into the 200 plus different cell types. Into the 200 plus different cell types. So if you think about how many types of cells are on your body in this little space that you have, stem cells are all about organization. And a key kind of characteristic of stem cells is progeny, or it's basically measuring the amount of potential a stem cell has to become these 200 plus different cell types. So at the top of this measure of potency, you have totipotent stem cells. Now you can probably guess what that means. It just means this stem cell has the ability to become all of the 200 plus different cell types. So everybody in this room, everybody walking the face of the earth has differentiated from one totipotent stem cell. Next, you have pluripotent stem cells. This includes your embryonic stem cells, which you can guess is a key aspect to the development of fetuses, and induced pluripotent stem cells. Next, you have your multipotent stem cells. That includes your hematopoietic stem cells, which are just blood cells, your neural stem cells, and a ton more. There are tons of multipotent stem cells. These multipotent stem cells have the ability to become about half of the 200 plus different cell types. Then at the bottom of this kind of pyramid of progeny, you have impotent stem cells, unipotent stem cells, and terminally differentiated cells. Is there any questions so far? Okay. So moving on. How do they function? What do stem cells do? Well, they are essential for the development of human tissue and regeneration of human tissue. So when you fall and scrape your knee in a soccer game or basketball game, stem cells are what's migrating to that area of injury and telling those cells already there to partake in the healing process. It's also differentiating into those kind of specific cells that are already there to make up for the lost tissue. So how do they function in the nervous system? Because that's what spina bifida is hindering, the development of the nervous system. Well, stem cells actually reside in this kind of niche or organized home in which they provide nutrients, support, and protection to that microenvironment. And what they do when triggered or activated in the case of injury, they migrate from this neuroepithelium or neural niche to the site of injury, and where they again tell other cells to start healing or to start replicating, and they themselves differentiate into the lost tissue. So now let's get into my paper. I've already established that surgery is not the definitive cure for neural tube defects because of the permanent and multiple disabilities that are effects of post-operative complications. So this paper is really addressing the matter of how can we make this method of treatment or this method of attacking spina bifida more efficient, because you have to realize spina bifida, although you probably don't know anybody who suffered from it, it affects a lot of people, a lot of babies. 1 in 2,000 babies worldwide have spina bifida, and 1 of 3 of those babies die before they see age 5. And the 2 out of 3 that do see age 5, they're looking at a life of constant dependency and a ton of complications that are testing their quality of life. So what's next? What's another method that we can think to kind of treat this defect? And this is where tissue engineering comes in. Tissue engineering is a combination of biochemical factors such as signals, of biological engineering material like scaffold, which can either be natural or synthetic, and live tissue, which are cells. So put that all together, and in the middle of this Venn diagram, you get tissue engineering. And it's basically just to regenerate the lost tissue as a causes of defects or diseases. And so they basically use this new and innovative method to achieve a better functional outcome for the current methods used today to counter spina bifida. So the first aspect of tissue engineering, cells. Which ones did they choose? They chose bone marrow mesenchymal stem cells because of its optimal use in labs. They self-renew, they're easy to isolate, meaning they're easy to work with, and they prevent fibrosis, meaning that they actually improve blood flow, which is crucial for cell health and growth, and it has less adverse reaction once placed into this organism. They're actually immunomodulatory cells, meaning they tell the immune system in the organism to not fight back against these cells. They're also multipotent. So the first step of this research experiment was to prepare the experimental subjects. They took 10 to 12-week-old pregnant rats and induced their embryos with spina bifida. That seems a little bit crazy, but it's actually really easy to do. You just take a driver of differentiation. In this case, they used retinoic acid to kind of hinder the development of these cells, and that's how they got spina bifida. Next, they isolated bone marrow stem cells from a separate group of 4-week-old Worcester rats, meaning they just extracted the stem cells from their bone marrow. Any questions so far? Yes.
You might not be able to speak to this, but does this retinoic acid treatment cause other neurodevelopment problems?
Yes, it does. Actually, I have worked with retinoic acid before. It doesn't necessarily have to affect the cell in a bad way. I use retinoic acid to differentiate just neural cells from regular stem cells, but in this case, they manipulated the procedure in which the cells would obtain spina bifida. I'm not sure how.
So it's probably localized.
Yes.
Targeted.
Any other questions? Okay, moving on. So the second step of this research project, was to isolate, to expand, and to transfect these bone marrow mesenchymal stem cells, or BMSCs. So they just cultured these cells in a lab with cell nutrients. In their case, they used DMEM-F12. It's basically just cell food. You have to remember that cells are living things, so when they took those cells from that 4-week-old rat, they immediately started to die, so they had to put it in nutrients. So this fluid is acting as that food and air for the cells. Then they supplemented this DMEM-F12 with 10% fetal bovine supplement, which is just more nutrients. And then they help these cells kind of prolong the cell health by adding penicillin. It's just an antibiotic because you have to remember bacteria is everywhere. It's on our hands, it's everywhere in this room, and so they had to protect these cells from getting infected. Then they cultured the BMSCs for expression. They're basically, they told the cells what to do, and this is them just double checking that they got the cells that they wanted. So they did this by performing a flow cytometry, which is basically a way of assaying the cells that you have in culture for markers that are found on the surface of BMSCs, which include CD34, CD45, CD90, and CD44. This flow cytometry was actually used as a purification assay. So how many of these cells had X amount of these markers, how many didn't, and so that's how they kind of got a level of kind of which cells were better than others. Lastly, they took their best cells and they transfected them with a fluorophore, or a fluorescent protein. They just incorporated this EGFP, the genetic material of this protein, into the DNA of the cell. This acted just as kind of a tag, so they can keep track of the cell and keep track of how many they had. This is a little technical, so everybody good so far? Okay. So next part of tissue engineering, I mentioned a scaffold or biological material. So in this study, they used chitosan gelatin for a number of reasons. Chitosan is it's a synthetic material, and it's known for its low biodegradability, meaning once placed in an organism, it wasn't going to break down or fall apart. They also chose it for its high biocompatibility, meaning once placed in an organism, it wasn't going to create an adverse reaction or be kicked out by the organism, as to say. Lastly, they chose chitosan because of its ability to produce porous scaffolds. You have to remember that tissue engineering is incorporating three main things: live tissue, signaling, and material. So these cells, these BMSCs, need a place to stay, and this scaffold, or spongy kind of material, is acting as a neural epithelium. It's acting as a stem cell niche. After they had grown this scaffold, they characterized it for its porosity and, more importantly, its size. Think of how small a human fetus is. Think of how small a rat is. Now put that together, think of how small a fetus rat is. That tiny break in the neural tube is extremely small. So they need to be able to fit as many pores as they need to fit the cells that they have cultured, and they need to be able to fit it in that gap. And so if one of these two characterizations don't come up to standard, then this entire process is going to fail. Any questions? So next step was to seed this scaffold with the labeled BMSCs along with more cell nutrients, so the cells didn't die. So here you can see this outline here, that's just the outline of the scaffold, and this green light is the cells that they loaded it with. They also stained, after the cells had a chance to grow in these scaffolds for 14 days, the scaffold along with the cells were stained. So it's really, really hard to see, but there's kind of red rings in this image, and that's depicting the scaffold. And then it's kind of a double perspective, so you can see that the red is in front of the green, and that just means that the cells successfully stayed in the scaffold. Any questions so far? Okay. So next step of this research project was implantation of the scaffold. Now this is just a disclaimer for you squeamish people out there. The next image is very graphic in nature, so I'm just asking you to brace yourself. So They combined a method of fetal surgery and microsurgery in order to effectively and successfully expose the defect region of the fetus. So this on the outer layer, this white fur, that's actually the body of the pregnant rat, and this is her fetus. Right here, you can see that little black dot, that's the scaffold. And so after they completed all the procedures, they actually found out that a ratio of 2 to 3 procedures went with no complication at all, no compromise of survival. So overall, the method of implanting this scaffold was successful. Next, 4 days after the babies, 4 days after the operation, the fetuses were harvested from their mother rats, and they collected the scaffolds for more re-examination and more assaying of the cells. So before they actually implanted this scaffold, they counted how many cells they loaded it with. And so this was kind of to see how many were still there, how many weren't, and whatsoever. And what they found was really, really remarkable. So 51 out of the 52 pregnant moms survived this procedure, and 69 out of their 134 fetuses also survived. And back to what I was saying about reassaying the scaffold, they actually found that the BMSCs that were loaded in this scaffold migrated and spread out across the entire defect region. So you can see these are the BMSCs labeled with the GFP still near the scaffold, and this huge cluster of cells that had moved out of it. It's also being shown right here. So this is just a immunofluorescence of the BMSCs. The red is the scaffold, like before, and the blue is actually cells that they activated in the defect region to partake in the neural healing process. So when you merge all of those images, you see that the majority of cells are actually outlying of the scaffold. So this scaffold proved to be an effective neural niche for these stem cells. Not only that, but this was the incision site of the fetus, and this is taken only 4 days after surgery. I don't know about you, but I've never seen someone fully recover from a surgical incision in 4 days. These stem cells also help to heal the incision site. So overall, this was a big A+ for this research project. You have to, even though there was only a 52% survival rate for the fetuses, this is ground zero for this new and innovative approach to attack congenital disease. So discussion. Do you guys think this new way of approaching congenital disease is effective?
You mentioned that the 69 fetuses survived, but that's just survival to day 4 post-op?
Yes.
So did they look at all at
They didn't follow these rats after.
allowing them to actually...
Yes, correct.
Kind of follow up on that. So they survived, and you showed the great data that the cells had started moving and differentiating. Did that fully make up for the defects, the scientific defects?
Well, that would be an inconclusive result. They would need to follow those fetuses up until at least prematurity, and they weren't followed 4 days after this post-op. They actually, in order to run all these tests, they had to sacrifice
But at
those
4 days,
fetuses.
did they look like a normal fetus or somewhere in between scientific and
I actually don't know the answer to that. I think that what they were just testing for is to see if they could improve the neural integrity, and so by activating the neural tissue that was already there to partake in this healing process, then that was itself is an improvement from just a fetus with regular spina bifida. Any other questions? Yeah.
You said like 69 out of 134 survived, right? Was there like a main reason that they didn't survive? Did they find it?
They didn't actually go into that, but I will look into that for you. I'm not sure. I can get back to you on that. I think it was just it had to do with the complexity of the procedure. And yeah.
Is the scaffold placed in that little neural gap?
Yeah, so if I go back to this picture right here, this is where they placed that scaffold.
Okay.
So it's extremely tiny, yeah.
So why is it a good thing if the cells leave the scaffold?
So, when I was talking about what stem cells do, one of their main jobs is to leave their neural niche, leave the place where they reside, to not only differentiate into lost tissue, but to tell the cells that are already there to start replicating or healing. So that's why they needed to migrate and kind of organize itself and distribute itself in the entire defect region. Any other questions? Okay. So, any other comments or questions about the discussion? What's actually really cool I found about this is that in the discussion they mentioned that spina bifida is correlated to anencephaly, which is the entire absence of brain matter in fetuses. So the baby's born without a brain. And so they were just talking about extrapolating this method for other congenital diseases. So I thought that was really cool. Any questions about that? Okay. Thank you guys for coming and listening to me present. Thank you to the General Club team, everybody who helped me.
So next week we have Miro presenting on his topic, which is the antibiotic apocalypse. So he's going to be talking about the superbugs.